We've seen in previous videos that photosynthesis can be broken down into the light-dependent reactions and the Calvin cycle. And the light-dependent reactions is where we take light as an input along with water, and we'll see the water is actually a source of electrons, and we can use that to store energy in the form of ATP and NADPH, and as a byproduct we produce molecular oxygen, which is very important for us to breathe. And then that ATP and that NADPH can be used in the Calvin cycle along with carbon dioxide to actually synthesize sugar. What we're going to focus on in this video are the light-dependent reactions. How does this process right here work? And to help us think about this, we are going to zoom in onto a thylakoid membrane. So this is a thylakoid right over here sitting inside of the chloroplast. And if we zoom in on its membrane, we see it's a phospholipid bilayer, like many membranes that we see in biology. And at first glance, this might seem like a very complex diagram, and that's because it is a complex diagram. And you will often see things like this in your biology textbooks, and it can be very intimidating. These proteins and molecules and complexes have very complicated sounding names, but the general idea of what's going on is you'll hopefully find pretty straightforward. You have the energy from light, photons from light, are going to either directly or indirectly excite electrons. Those excited electrons, they're in a high energy state. They're going to be transferred from one molecule to another, and they're going to go to lower energy states. That's what allows those transfers to be spontaneous, for them to actually occur. They're going from a high energy state to a lower energy state. The electrons are getting more and more and more comfortable, and some of that energy that's released as the electron goes from a high energy state to a lower energy state is used to pump hydrogen ions across the membrane, from the outside of the membrane in the stroma to the inside of the membrane, to within the thylakoid lumen. So you're building a hydrogen ion concentration gradient. Concentration gradient, where you have a higher concentration inside than you have outside. And this by itself, this concentration gradient, as we'll see, can be used to fuel the production of ATP by ATP synthase. That those hydrogen ions want to get back out. They want to go down their concentration gradient, and as they go back out through the ATP synthase, it essentially turns that motor that can jam the phosphate group onto ADP to produce ATP. So one way to think about it, this is producing a hydrogen ion gradient. So we could do it this way. We could say H plus gradient, which is then being used to produce the actual ATP. Now, the electrons going from a high energy state to a lower energy state in this part of the light-dependent reactions, that by itself isn't the only thing that is contributing to the high hydrogen ion concentration gradient. Once that electron gets donated, you might say, well, how does it get replaced? Well, the thing that's doing the donating, the thing that eventually gets excited and donates that electron, it's a chlorophyll A variation called P680. P680 is referring to, the P stands for pigment, 680 stands for 680 nanometers, the wavelength of light that it absorbs best. And so when it gets excited, it becomes, you'll see the notation often of P680 star, that's when it has an excited electron. And then after it gives away its electron, it becomes P680 with a positive charge. And this P680, we could call it P680 plus right over here, or maybe a P680 ion. This is actually a very strong oxidizing agent. One of the strongest, if not the strongest, we know in biological systems. And so it really likes to grab electrons from other things. And the thing that is around that it can grab electrons from is actually water. And so this is such a strong oxidizing agent that it can essentially oxidize the oxygen in water. And oxygen is itself, oxidizing is named after oxygen because oxygen is such a strong, it's so electronegative, it's such a strong, it's the thing that's normally doing the oxidizing. So anyway, it grabs its electrons. Once it gets this P680 plus, grabs an electron from water, and then the water essentially falls apart. So you're left just with the oxygen and then the hydrogen ions. And so those hydrogen ions also contribute to the increased hydrogen ion concentration on the inside. And this is where we get the oxygen byproduct right over here. Here we have 1/2 of an O2, so if you do this twice, you're going to have a molecular oxygen. So far, we've talked about how the oxygen gets produced. We've talked about how the ATP gets produced. What about the NADPH? Well, we started our process in photosystem 2. You might say, why is it called photosystem 2 if that's where we start? Well, that's because that's the second photosystem to be discovered. You might say, what is a photosystem? Well, these photosystems and complexes, they're combinations of proteins and molecules, and photosystem in particular has chlorophyll and variations of chlorophyll and pigment molecules that are responsive to light, that are very easy, that have electrons that can get excited by light, and they can transfer that energy back down to the P680 chlorophyll A pair, which then can have its electron excited, and then it can give that to an acceptor molecule, and then it can go to lower energy states and pump those hydrogen ions out. But that's not the entire light-dependent reactions. That electron can eventually make its way over to photosystem 1. And why is it called photosystem 1? Well, it's because the first one that was discovered. In photosystem 1, there's another chlorophyll A pair called P700. And that's because it optimally absorbs light of a wavelength of 700 nanometers. And you have something similar that happens, that light can either directly or indirectly excite its electron. And then that electron, as it goes to a lower energy level, it goes from one molecule to another, it can be used to reduce NAD plus into NADPH. And so that's where the NADPH comes from. And then once again, once the P700 has given its electron, it wants an electron, and well, it can get that from the electron that's been going from lower to lower and lower energy states, that's essentially been making its way from, you can conceptualize it as the electron that's been making its way from photosystem 2. And so that's why you'll often see these diagrams. Lights come in, electron gets energized, it gets excited, it goes to lower and lower energy states. As it's doing that, it's being transferred from one molecule to another, being facilitated by enzymes. That energy, part of that energy, is being used to transfer hydrogen ions into the thylakoid lumen, into the interior. Then in photosystem 1, you have another excitation event. That thing that got excited can grab that electron that went to lower and lower energy states, and its excited electron can once again be transferred from one molecule to another in order to fuel or provide the energy for NADP plus being converted into NADPH. And once again, the whole idea of the hydrogen ion concentration increasing here can fuel ATP synthase, which allows us to jam a phosphate onto ADP to produce ATP. So that is where we actually get all of these things, and the byproduct, of course, is our oxygen. And if you wanted to see that same idea, but kind of just thinking from an energetic point of view without all the complexity of seeing the physical components involved, you see it right over here, where you have light energy comes, excites the electrons. Once the P680 has given that electron away, it wants an electron really badly. It gets it from the water. And then as that electron goes to lower and lower energy states, it can eventually be grabbed by P700 that has given away its electron. And then that electron that was excited at P700 by, once again, more light energy, that can be transferred from one molecule to another to fuel the creation of NADPH. And this part right over here, this phase right over here, as the energy goes from a high energy state as the electron goes from a high energy state to a lower energy state, fuels the pumping of hydrogen protons into the actual thylakoid.